Read back nanti. At one point, I akan bagi balik your lab report just for you all to go through for next year lah. At least you orang tahu kat mana salah silap and so on lah. Okay. That day we just isolate ataupun extract the DNA. Kita tak buat PCR. PCR tu untuk memperbanyakkan DNA. Dia punya sequence tu. Okay. So kita tak buat benda tu sebenarnya. Okay. So let's see how we can go about it. Today I'll try to explain a bit on that. And I hope you'll be okay lah. Okay. You all will try to understand. Dah macam molekular kan. Sekejap I nak buat macam mana ni. Admit. Okay, sekejap. Cancel. Kalau dah pukul 1 tu nanti stop ni. You ada kelas pukul 2
Pukul 3 doktor.
Okay, sempat dah habis ni. Dengan Dr. Zarina kan? Okay. Okay, sekejap. Can see Okay, so our topic today is related to your lecture, on your lab yang hari tu, on the molecular biology. So we will learn on DNA, what's DNA. I think generally you guys know what's DNA theoretically. Some part of it is very basic, what you have learned in Form 5 and maybe Form 6 or matriculation, and then followed by techniques of PCR lah. Okay, as I told you, we didn't do lab on PCR, we just isolate the DNA. So today I will brief you on PCR method, which you might learn better next semester or the following semester. Okay. So what is a DNA? You orang semua tahu ni what is a DNA? It's a nucleic acid. Okay, nama dia adalah deoxyribonucleic acid. DNA is a polymer of a deoxyribonucleotides. Okay, polymer kalau it's a lot of nucleotides, deoxyribonucleotides combined together. Usually it's a double-stranded. Okay, nanti kita tengok what's a double-stranded DNA. And have a double helix structure. It is found in chromosomes, mitochondria, and chloroplast. So these three organelles, it will have a DNA, chromosome, mitochondria, and chloroplast. It acts as a genetic material in most of the organisms. Okay, genetic materials. Okay, carries the genetic information. Okay, it has genetic information, it has genetic material which carry our genetic information, the living things. Okay, I think this is very, very general lah. You know who found it and James Watson and so on. I'm not going to go through this. What is the purpose or function of DNA? It stores the genetic information that instruct the cell on which protein to make. So every function in a living things is controlled by DNA. How it does that? This DNA, as you know, it will transcribe to a protein. Protein will be a message bringer. It will be a mRNA, it will bring to protein, it will convert to enzyme, and so on. It has a lot of function. Okay, if we go very in deep molecular biology, you will try to understand everything, every part of that. Okay, responsible for determining all organism traits such as for us, eye color, body structure, enzyme production, blah blah blah. Okay, and understanding individuality of a plant using genetic information of DNA for plant modification. So if you, as I told you, if you want to modify a plant, its trait, you have to do through its DNA, its molecular studies. Okay, So proteins, bukan protein yang kita makan tau. I mean like memanglah protein tu juga, tapi protein kita makan yang besar kan. So ini proteins maksud dia amino acid dia yang converts. Okay, this kind of proteins. Okay, proteins are responsible for most of these traits. Okay, in all these characteristics of a plant and so on. Okay. DNA, okay, this one is a bit more in detail, which you need to know. DNA structure is often divided into 4 different level. 4 different levels, which is primary structure, secondary structure, tertiary structure, and quaternary structure. Okay, so we go through one by one. The components of DNA, yang ni orang pun dah pernah belajar. DNA is a long molecule made up of repeating individual unit of monomers called nucleosides. So nucleotides. So this is the nucleotides. Nucleotides ada 3 parts that are held together by covalent bond, the bonding. Okay, so it has a deoxyribose, a pentose sugar. It has a phosphate group. It has a nitrogenous base. Okay, ATCG. Okay, that's a base of a nitrogen here. So this one is the one that makes up nucleotides. Nucleosides are similar to nucleotide but have no phosphate group. So this only the hexagon dengan yang hijau ni combine, it is called nucleotides. This one you all know. Okay, and nitrogenous base, what is this? Adenine, thymine, cytosine, guanine. Okay, dalam DNA dia ada ni. Okay, this is the different lah. It differs from one to one. Okay. Okay. The pentose 5 carbon sugar is in RNA is ribose. Okay. In RNA kita panggil dia ribose. DNA is deoxyribose with no oxygen atom on carbon number 2. So this is the structure, it's the molecular structure. This is the pentose part. Okay, what's the difference between like ribonucleic acid, ribose, and deoxyribonuclease? Kalau DNA dia ada hydrogen group dekat carbon number 2. Yang ni ada hydroxyl group. Okay, dekat carbon number 2 ribose. Okay. As carbon atoms numbered with primes to distinguish them from the atom in the nitrogen base. Okay. Okay, DNA contains 4 nitrogenous bases that are adenine, guanine, cytosine, and thymine. Okay, this is adenine structure, this guanine structure, cytosine, thymine. So usually they bond together over here from one to another. Yang ni bond dengan yang dia punya phosphate group, dia punya pentose sugar dengan phosphate group. So it makes the chain longer. And purines and pyrimidines, the difference is here. Okay, nanti kita tengok what's the difference. Okay, this is adenine dengan guanine are purines. Adenine dia match dengan thymine, guanine dengan cytosine. Okay, dalam dia punya alpha helix tu. Okay, so purines and pyrimidines. So purine kita ada adenine, guanine. Pyrimidines dia ada cytosine, thymine, uracil. Okay, pyrimidine, purine, it has 2 carbon nitrogen rings. Okay, carbon nitrogen rings, 4 nitrogen atoms. Okay, pyrimidine, it has 1 carbon nitrogen ring, 2 nitrogen atoms. It has 2 nitrogen atoms. Yang ni ada 4 nitrogen atoms. Dia ada 2 carbon nitrogen ring. So these are the differences, main differences lah. Okay, selain daripada dia punya in detail differences. Okay. So nucleoside and nucleotide. Nucleotides are formed by the condensation of a sugar, phosphate, and one of the 4 bases. Okay, sugar, phosphate, one of the 4 bases, dia condense, dia akan produce nucleotide. Kalau nucleoside aja, sugar dengan base akan condense forming nucleoside. Okay, condensation reaction dia berlaku for phosphate, adenosine, dia bertambah phosphate dengan adenosine is a sugar here, dia punya group lah, adenine dengan ribose, adenosine. It condense forming a adenosine monophosphate. Okay. Okay. Base plus sugar is nucleoside, sugar ni. Adenine plus ribose is adenosine. Adenine plus deoxyribose is deoxy adenosine. Base, sugar and phosphate is nucleotide. Okay, 3 things is a nucleotide. Example is deoxyadenose monophosphate, deoxyadenose diphosphate, deoxyadenose triphosphate. Okay. Names of nucleosides and nucleotides. Okay, these are different names lah. I don't expect you to hafal lah, but the long names you don't need to hafal. Usually kita panggil dia adenine, guanine, cytosine, uracil ataupun thymine. Yang long name ni, it's not very important for you to remember anyway. Okay. Jadi kita tengokkan dia punya structure ada primary, secondary, tertiary and quaternary. Okay, primary structure of a nucleic acid is the general insight lah how it is. So it develops like dari kecil jadi besar kan, macam how it tied together is the structuring punya ni. Primary structure of nucleic acid. In primary structure of nucleic acid, nucleotides are joined by phosphodiester bonds. This is primary structure of nucleic acid, nucleotides lah. Nucleotides are bound by phosphodiestic bonds. So kat sini dia connect. This is one nucleotide, this is another nucleotide. Kan? This is one, this is another. So the connection dia adalah dekat sini, tempat ni. Yang I tulis phosphodiester bond. Okay. The 3' OH group of the sugar in one nucleotide forms an ester bond to the phosphate group on the 5' carbon. So 3OH ni dekat 3, dia ada OH ni, dia bind dengan 5th carbon dekat phosphate ni of the sugar of the next nucleotide. Okay. Nucleotide residues are all orientated in the same direction, 5' to 3', giving the polymer directionality. Okay, this is from 5' to 3'. Okay, the sequence of DNA molecules is always read in the 5' to 3' direction. So when you do molecular biology, they will ask from 5' to 3'. That's how they read it. Okay, nucleotide tu macam A, G, C, T, T macam ni kan. Dia punya read, dia mula daripada 5' to 3'. Maksud dia punya combination dia macam mula, dia punya basis dia adalah thymine T, A, C, G macam tu. Dia akan mula read in a sequence of a DNA. Okay. Okay, DNA primary structure. The primary structure of nucleic acid is the linear sequence of nucleotides. It's a linear sequence of nucleotides. Okay, this is where it connects. Okay, which are connected to each other by phosphodiester connections. This is the phosphodiester connection. This is how it is connected. Any next? Okay, this is one. This is one. This is another one. This is what I was telling just now as well. And this is where it starts the 5'. This is where it ends the 3' ends. Okay. Okay. Tadi kita dah tengok how the components function. Components dia ada dalam ni tau. Dalam tempat kecil-kecil ni. So when all the component macam benang ni, mula-mula dia macam benang. Sekarang kita kena tengah buat kain yang besar sikit. Faham tak? Kain yang 2 layer lah, macam kita tengah buat kain. Lepas tu kita akan buat baju. Macam tu lah. So yang primary structure tu macam benang je. How do you make a thread? Now you are making a cloth. Bukan cloth. You are making a kain dululah. Kain biasa. Kain berjala tu kan. So you connect all together. So who discovered the secondary structure of DNA is Watson and Crick. Watson and Crick discovered the secondary structure of DNA. A double helix 2 sugar phosphate chain wrapping around each other with the phosphate group sticking out. So this is how it is. This is one structure kan. Kita tengok kan dia punya helix kan, dia macam pusing kan macam ni. Dari sini pusing, sini pusing. So then dia connect, tempat yang dia connect adalah a double helix of 2 sugar phosphate. Yang double helix of 2 sugar phosphate tu, tempat yang kat 2 ni dekat sini yang attached to satu benang dengan benang yang lagi satu. Dia macam satu benang dengan benang lagi satu, dia twist as a Watson Crick lah. Okay, round each other with the phosphate group sticking. The nucleotides from strand 1 meets the nucleotide from strand 2. Okay, nucleotide yang jumpa dekat ujung-ujung ni. Nucleotide ni dengan nucleotide ni berjumpa. Okay, these pairs of nucleotide are complementary. So in a benang tu yang jumpa tu adalah nucleotide, tapi what dijumpa dia ada 4 jenis saja kan nucleotide. So adenine will pair with thymine, guanine will pair with cytosine. Okay. Any question? Hello.
No.
So far benda ni semua orang dah pernah belajar kan somehow. Bila?
Genetic.
Genetic. So this is just adding lah. So there will be I think two questions from apa daripada your this topic in your finals. Okay. I think two. Okay. The DNA double helix secondary structure. Okay. This is the secondary structure, how it bonds lah. The base in the two strands are paired through hydrogen bond. So the bonding, ini kan satu pair kan yang tadi dia lipat-lipat tu kan dah buka dah. Mereka dah buka. Okay, dah buka. Kalau dibuka dia nampak macam ni lah. So dia punya bonding yang ni satu pair, satu benang tu yang primary structure tu dengan benang yang lain satu tu dia di pair dalam hydrogen bonding. Adenine is paired to thymine, guanine is paired to cytosine. Okay, so yang ni yang kita kata, yang ni dia mula 5' to 3' dekat bawah. Yang ni yang atas ni 5, yang hujung ni 3. Yang ni opposite, dia akan 5' kat bawah until 3' kat atas. Okay, that's the direction. The base in two strands are paired through hydrogen bonding forming base pairs. Kita panggil dia base pairs. Yang ni kita boleh kata 1, 2, 3, 4, 5, 6 base pairs saja. Okay, dia ada 6 base yang pairing. Okay, itu yang kita panggil BP. Okay, adenine forms 2 hydrogen bonds. Adenine 2 hydrogen bonds with thymine from opposite strand and vice versa. Similarly, kalau yang ni bond G dengan C lagi strong kerana dia ada 3 hydrogen bond. Kalau AT dia ada 2 hydrogen bond aja untuk pair mereka. Okay. Okay. Based on the observation of Erwin Chagav, that for a double-stranded DNA, the ratio between adenine and thymine and guanine and cytosine are constant and equal ones. Hydrogen bond. A chemical bond consisting of a hydrogen atom between two electronegative atoms. Okay, example, oxygen or nitrogen, with one side be covalent bond and the other being an ionic bond. Okay, hydrogen bond lah. It's quite in detail. Okay, this is the hydrogen bond. Okay, between thymine and adenine, between cytosine and guanine. Okay, one is pyrimidine, one is purine. I think this is purine, this is pyrimidine. Okay. Okay, as I told you, DNA strands are antiparallel. One strand runs 5' to 3', as I told you. Another strand is 3' to 5'. Okay. DNA strands are twisted to form the double helix. That's what yang Watson and Crick find as a secondary structure lah, double helix dia. Okay. Okay, H bonding of adjacent antiparallel DNA strands form double helix. So, yang ni yang pairing kan, yang ni let's say this is thymine, this is adenine, yang ni guanine, yang ni cytosine. So, when it pairs, dia join, lepas tu dia akan twist. Macam ni. Okay, this is 5', it ends dekat 3'. So, you see kalau dia akan end dekat 3'. So, 3' and at 5'. So, it's very simple things. Okay. The two strands are coiled in a right-handed fashion. Okay, right-handed. Alamak, apa jadi? Ouch, ouch. Okay, right-handed. Right-handed fashion. Clockwise, like it moves clockwise. Okay, double helix DNA turns after every 10 base pairs at the distance of 3.4 nanometer. So, from here, it turns back at the same position. Lepas setiap 3.4 nanometer, dia akan pusing direction yang sama. Okay, macam twister you lah kan. Twister ke, bintang twister yang boleh. At one point. So, dari tempat yang sama, dia mula nampak dalam 3.4 nanometer. Okay, two base pairs in a helix approximately. This distance is called pitch. Consequently, the distance between two base pairs in a helix is approximately equal to 0.34 nanometer. Here 0.34. Here 3.4. Okay, yang gap dia. Okay, macam bagi 10 lah. The plane of one base pair stacks over the other in double helix. This is in addition to hydrogen bond, confer stability of helical section, blah, blah, blah. This is structural molecular biology, okay? Okay, there are two asymmetrical grooves on the I side. Okay, ini macam groove aja lah. Ini minor groove, yang kecil-kecil groove, yang space dia. Ini major groove. Groove kalau yang hole kat sini lah. Tak penting sangat pun orang nak tahu. Minor groove, major groove, okay. Tak ada apa pun benda ni, okay. Ini dia bagi size structure lah. You no need to know very in detail lah. Nampak pun dah tahu dah macam one complete turn is 3.4 nanometer. Ini small turn, ini ada groove kat. Macam tu lah. Okay, ini major groove, yang ni minor groove, small. Okay. When he proposed, dia tak bagi, okay, he proposed few different types of structures, Watson Crick ni. I mean, not Watson Crick lah, few of them propose how it looks. So structures of for a number of synthetic oligonucleotides 4 to 24 self-complementary bases, 4 to 24 bases that folds into a double helix have been determined under various conditions. This is reveal the basic structure of three fundamental types of double helix. So how it looks, Watson Crick punya dipanggil dia B-DNA structures, how it folds. Ada yang A, ada yang Z DNA. So what are their difference? This is what other forms of DNA have been crystallized, including B prime, C prime, blah, blah, blah. All of these are right-handed structure and occur under unique conditions. For example, cDNA forms in the presence of lithium salt and low humidity. So Z ni lain. Okay. Yang kita punya yang normal tu adalah B, which is Watson and Crick punya model lah. This is the B yang kita ada. So at the high humidity 95, low salt dalam body, normal salt, your DNA will be like this. If it's a high salt, low humidity, it will be like this. Okay. If left-handed, okay, in the presence of methylated cytosine, high humidity and low salt, it will look a bit like this. Okay. Which is a different structure, a different condition. Okay, chemically it is being affected. Okay. But still, it is still a helix lah. Okay. Okay. DNA replicate. How do they produce? Kita pernah belajar kan meiosis, mitosis and so on kan. How do a cell replicate? How do a cell make it a lot? Okay. The process by which DNA make a copy of itself is called a cell division kan. So this is the parental cell ataupun old cell. Then dia akan unzip, dia akan buka, dia ada double helix, unzip the new bases. Then two new strands are created. Each contain half of the original strain. So the same strain akan mula. Untuk buat proses ni akan ada enzyme and so on lah. So you can dapat dua. This is mitosis because it is forming a identical thing. Just it's duplicating, becoming a identical DNA. Okay, cell division. Okay. Excuse me. Before a cell divides, DNA untwist and unzip down the hydrogen bond. This is why the bonds had to be weak. So the bond is not very strong. Okay, dia ada dua hydrogen atau tiga hydrogen bond. So dia akan break open. These strands make a new partner by adding an appropriate nucleotide. So dia akan add lah. Kalau yang ni A, dia akan add T. Kalau yang ni C, dia akan add the pair will be a G. Macam tu lah. Dia akan add, dia akan form a new one. The result is that there are now two double-stranded DNA molecules and the cell division process will continue. Okay. What happens when there's a problem during replication? If mutation happen, if in human, that is the one that causes cancer lah. There's some changes in the DNA sequencing. Ataupun kalau dalam plant, we grow callus out of this mutation. It changes the DNA sequence. A permanent change in the sequence of DNA is known as a mutation. A small change that occurs during replication but is caught corrected is known as a point mutation. So satu lupa apa? Satu point pun akan ada issue. Okay. Okay, so you see, so correct copy, ini yang original. So ini jadi mutant. If you see here, it opens kan. So this is the original. When it opens, okay, it connect back. But here, it becomes one thing salah. Ini yang original kan. Ini yang one thing dah salah, then it makes a total different thing. Okay. Denaturation of DNA. Denaturation means you unwind ataupun you buka daripada double helix jadikan dia straight, separate the DNA strand. This known as melting lah. Kalau PCI kita panggil dia melting. Dia denature. Dia akan untuk buka, untuk buka benda ni kan, daripada ni nak buka je kita panggil dia denaturing. Bukan. Denature lah. H bonds between the H pairs can be broken and DNA strand separated by heating the DNA molecule. Covalent phosphodiester bond remain intact. So you denature je, buka je. Temperature at which half of the base pair in a double-stranded DNA sample have denatured is called the melting temperature. Half of the base pair. So kita panggil dia melting temperature TM. Okay. Excuse me. The higher the GC content in a given molecule of DNA, the higher the temperature required to nature the DNA. Okay. So when the GC content is higher, because GC content dia ada more, dia ada 3 hydrogen bonds. So kalau dia lagi tinggi, dia punya temperature lagi tinggi kerana dia kena break 3. Okay. Lowering the salt concentration of DNA solution promotes denaturation by removing the cation that shields the negative charge on the 2 strands from each other. So if you want to denature, you can also do the lowering the salt concentration. At low ionic strength, the mutually repulsive forces of these negative charges from the backbone phosphoryl groups are enough to denature the DNA, even at a relatively low temperature. High pH or organic solvents such as formamide disrupt the hydrogen bonding between DNA strand and promote denaturation. Okay, high pH pun lah. Okay, this is how it happens lah. This denatured and then hybridization. Okay. Hybridize is you form another new one. You masukkan yang baru. When heated solution of denatured DNA are slowly cool, dia akan form balik, bond balik. Single strand often meet their complementary strand and form a new double helix renaturation or annealing. Okay, so dia akan anneal balik, dia akan form balik. The capacity to renature denatured DNA molecule permits hybridization, the complementary base pairing of the strand from 2 different sources. Okay. Okay, now kita dah tengok satu benang, 2 benang. Benang satu benang adalah primary. Kalau ada dia complementary, dia ada secondary kan. Okay, yang ni tertiary, dia macam akan twist together lah. Okay. Many naturally occurring DNA molecules are circular with no free 5' or 3' end. Why? Mostly dia akan bentuk circular kerana dia mudah. Kalau circular dia boleh twist turn and simpan banyak. DNA ni panjang, dia tak boleh simpan panjang kan dalam body. So dia akan twist and coil to reduce the space. Due to the polarity of the strand of DNA, helix, the 5' end of one strand can only join its own 3' end to covalently close the circle. So its own lah dia akan close balik. Okay. Circular double-stranded DNA is essentially 2 circle of single-stranded. So, kalau you tengok kat sini, mungkin dia mula dekat sini as a 5'. Dia pusing je tu, dia habis kat sini 3', so dia connect back. It's a circular bulat double-stranded DNA is essentially 2 circle of single-stranded DNA twisted around each other. This DNA can then become supercoiled twisted. 3D structure which is more favorable energetically can be visualized by electron microscopy and occurs both in vivo and in vitro. Okay, one helical turn is almost 10.5 base pairs. Okay, base pairs means ini satu base, 2 base, 3, 4, almost 5. Untuk satu turn dia ada 10.5 base pairs. Okay, this is how you see the base pairs. Okay, this is the tertiary structure. Dia ada coiled up. You tengok, dia mula dari sini, dia pusing, dia masuk balik. Dia coiled up as a circle. Ini pun dia coiled up. Okay, that's a tertiary structure. Okay, DNA supercoiling refers to the over or under winding of the strand. The supercoiled state is inherently less stable than relaxed DNA. Okay, dia less stable, dia macam dah coil sekali, tapi dia less stable, so dia boleh buka balik juga, jadi benang balik. Okay, bukan benang lah, yang pair of benang DNA tu. DNA supercoiling is important for DNA packaging within all cells because the length of DNA can be thousands of times that of the cell. So, dia boleh panjang sangat, so dia kena kumpul. Macam you letak benang kan, you tak boleh lah buka benang tu panjang-panjang kan, you kena gulungkan macam itulah, dia gulung sendiri. That of the cells packaging this material into a cell or nucleus is a difficult field. Supercoiling of DNA reduces the space and allows for much more DNA to be packaged. Banyak lagi. This has important implication for cellular process such as replication and transpiration. Okay, this is how it is called. This is the DNA. Kan tadi kita tengok ini yang primary structure dah, yang ni dah secondary structure. Okay, Watson and Crick, then dia coil together lah jadi benang and so on, jadi secondary, jadi tertiary, okay, jadi quaternary, and then masuk dalam your chromosomes. Okay, this is our human chromosomes kan. Okay, okay, this is on the quaternary structure. Nucleosomes are the basic unit of chromatin organization. Okay, nucleosome lah. Okay, in eukaryote DNA associated with protein. In prokaryote DNA is naked. Naked means dia tak ada lah protein and so on kan dalam prokaryote. Yeah, I think this is not very important, but we just skip this, we go to this one. But give me like 2 minutes and let me know how you do. Okay. You all okay ke? Too fast ke? Ataupun memang benda yang you orang dah tahu dah?
Okay.
Okay, Doktor.
Ya, pergi baca balik lah kan. Lagi 2 orang tak ada tak sempat nak join ke apa?
Rasanya terleft tu.
Terleft. Okay, tak apa. Ask them to read. Okay, so sekarang yang I nak, what I want to lecture you on what is PCR. Okay, I've told you earlier what is a PCR actually. So DNA ni, your report, I told you buat tak ada kena-mena dengan PCR. You didn't go to the PCR. Of course, you discuss about PCR, so that's what I'm coming here. So once you have already isolated yang, you isolate your DNA dah macam ni dah. All the DNA dalam leaf tu you dah isolate. So dalam PCR you nak, yang ni all the genes, all the sequence you dah isolate. So baru you masukkan dalam PCR. Dalam PCR tu kan tadi kita baca kan every sequence ada dia punya sequencing. 5', 3' kan, dua belah tu you akan ada 5' and 3'. Macam ni kan, this part and this part yang I tak share. Sekejap. Ini cakap tapi tak share. Kita nak belajar PCR kan. So what you will do in PCR, okay, you will do this kind of process. Okay, you have a DNA, itu you isolate the entire DNA of the leaf. So you nak satu sequence saja. Okay, you nak specific sequence. Okay, for the leaf, in making the leaf, what to say, the leaf cells to look ataupun leaf has that compound. You nak satu gene yang responsible for the compound. Satu je gene. Okay, kena kita extract, kita isolate semua, the entire gene. So you nak satu je. What you can do is, if this is the gene tu perlu dibuka and you have to do the PCR. So when you open this thing, you ada gene sequence, you akan ada yang primer. Kita panggil dia primer. Primer adalah benda yang mula. Kita kan tadi belajar kan 5' to 3'. Every gene will have a, I cari balik. Mana dia tulis 5', 3' tu. Okay, 5' to 3' kan. So, you dah isolate this entire gene. You nak satu gene je. Yang ni satu gene. 5', dia end dekat 3'. Ini 5', dia end dekat 3'. So, you buka benda ni, you open it, jadi dia akan jadi macam ni when you open it. Okay, when you twist, it's like that lah. When you open this, the gene, you kena buat sequencing dalam PCR tu akan memperbanyakkan gene ni. Macam mana dia akan buat? Dia akan buka benda ni, lepas tu you dah kena ada tahu dah sequence tu dia punya starting apa dia punya nucleotides. Dia punya sequence tu kita buat, itu yang kita panggil dia primers. Dia akan buat dekat sini sikit, tempat ni dengan kat bawah ni. So dia akan mula kod lah, dia akan pergi, you ada primer tu, dia akan pergi lekat. Primer tu akan pergi lekat dekat sini. Primer, forward primer kita panggil dia, yang ni reverse primer, dia akan lekat dekat sini. Dekat sini dia akan baca, dia akan baca, and then dia akan buat lagi satu strand. So dia akan buat macam ni. Dia akan buat macam ni. So dia akan baca, lepas tu dia akan buat naik panjang lah. Dia akan buat dua. So yang ni dua dah jadi berpisah kan. So dia akan berpisah, dia akan buat strand. Ini akan buat strand lagi. Benda ni akan repeat lah. Benda ni pun akan buka dan dia akan buat lagi banyak. That is what PCR is about. Okay. So that day kita isolate je DNA. Okay. So now we go to PCR. So generally what is PCR is that lah. Okay. Sekejap. I share yang besar sikit. Okay. Okay, PCR is a technique to rapidly make millions to billions of copies, complete or partial, of a specific DNA sample, specific gene, specific DNA sample. Why do you want to do millions to billions? Kerana kalau sikit, you cannot detect, you cannot baca benda tu. Kena dalam plan mungkin ada satu dua je. So you banyakkanlah dia. Dia replicatekan dia. Allowing the scientist to amplify means jadikan banyak specific DNA primer. It uses a very small amount of template. Okay, small amount of template. Utilizes a heat stable DNA polymerase. Okay, dia need enzyme to make the make a lot of new DNA tu untuk replication lah. Okay, from bacteria living in hot spots or hot springs. So DNA tag polymerase is from bacteria. Kita ambil dia punya polymerase untuk buat DNA tu. Uses repeated cycles of denaturation. Tadi kita belajar kan what's denaturation means. Yang coil tu you pergi buka, DNA replication, lepas tu you buka, lepas tu you replicate dia jadi banyak, and then cooling. You replicate jadi banyak, lepas tu you cool balik kerana you nak dia twist balik jadi DNA balik. Okay. Cooling to double the amount of DNA during each cycle. Uses an automated thermocycler. Yang inilah mesin PCR ni. Nama mesin PCR ni sebenarnya adalah thermocycler. You boleh set the temperature and so on. You boleh letak dekat dalam ni. So dia akan ada 3 proses. Okay. Dia akan ada denaturation, annealing, and extension. Okay. Nanti kita tengok one by one. Okay. PCR primer is the one I told. Depan dengan belakang yang sequence pendek tu. PCR primers are short pieces of single-stranded DNA, usually around 20 nucleotides in length. Okay, 2 primers are used in each PCR reaction and they are designed so that they flank the target region that should be copied. Okay, yang ni region that you want to copy. Ini yang template DNA kan. Sekarang template DNA yang coil tu you dah buka jadi straight, tapi dia still bond, ada bond. Lepas tu you buka dan nanti dia denature, bond tu akan terbuka. So you design lah primer, ini yang 5' to 3'. So you buatlah satu kat sini primer, lagi satu kat atas ni primer. So dia akan complementary, dia akan buat 2. Dia akan buat, lepas tu benda ni akan buka balik, dah jadi 2 kan, benda tu akan buka balik dan proses yang sama akan berlaku, making it thousands to millions to billions amount of the same region. Okay, region that should be copied. They are given sequence that will make them bind to the opposite strands of the template DNA, opposite strands DNA, just at the edges of the region to be copied. Okay, this is the primers lah. Okay, thousands. Okay, the primers bind to the template by complementary base pairing. So kalau this is the base pair, you kena design a primer that is complementary. Macam dia kata kat sini kan, let's say kat sini. Okay, dia ada ATA, GTC, bla 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 kan. So G punya ni, you nak design primer kat sini, G punya base, primer dia must be C, A, G, macam complementary bases lah. Okay. So this is an example. Okay. When the primers are bound to the template, they can be extended by the polymerase, and the region that lies between them will get copied. Okay. So primers bind to the template, and then it can get copied. Tag polymerase extend the primer. So polymerase ni akan tolong. Polymerase is an enzyme kan, dia akan membantu untuk dia jadi panjang. So how do you get their base? Kita akan supply, kita panggil dia nucleotide by ourselves. Masa kita nak buat reaction tu, sebelum kita letak dalam mesin ni, kita akan supply dia dengan enzyme, kita akan supply dia dengan sequence tu, yang adenine, guanine, cytosine. So dia akan pergi lekat lah dengan complementary bases tu dan mula jadi panjang guna enzyme tu. Okay, this way and this way. Benda ni akan repeat lah. Yang ni jadi 2, 2 jadi 4, 4 jadi 8, 8 jadi 16, macam tu lah. Okay. Okay. Kita ada 3 steps of the PCR reaction yang I kata tadi. Kalau kita letak dalam mesin tu kan. So, kita dah masukkan semua dalam satu tube. Kita akan masukkan template DNA kita, DNA asal kita. Kita akan masukkan primers, forward, reverse. Kita akan masukkan enzyme yang I beritahu tadi. That is a polymerase enzyme. Lepas tu kita akan masukkan nucleotide yang strand lah. A, C, G, T tu kita akan masukkan secara asing kat dalam. Lepas tu kita proses semua, kita letak extra water ke apa ke, everything according to the manual. Lepas tu kita letak dalam PCR machine. Dalam PCR machine apa yang berlaku adalah, proses adalah, first adalah denaturation. Dia akan, daripada 2 benang yang coil tu, dia akan buka dia punya bonding. Okay. The targeted sequence of DNA is edited to denature the template strands, untuk buka and render the DNA single strand. So dia akan jadi 2 strand yang asing, tak ada bonding. Okay. Lepas tu annealing. The DNA at the appropriate complementary strand. So dia jadi 2. You jangan temperature dia tak boleh tinggi sangat. You nak attach primer je. So primer punya annealing temperature je. So dia akan ada primer kat sini satu, lepas tu lagi satu pun kat bawah ni. So dia akan boleh primer tu melekat. Okay. The temperature for these steps varies depending on the size of the primer, the GC content and its homology to the target DNA. Primers are generally DNA oligonucleotides of approximately 20 bases each. So this is the process. Handling is for you to combine the primers dulu. Okay. Lepas tu baru dia extend. Extend means this is the sequence. You dah ada ini yang primer yang pergi lekat kan. Lepas tu dia akan mula extend. Primer tu akan tambah sampai dia form the whole DNA sequence. In the presence of magnesium ion, DNA polymerase extend the primers on both strands from 5' to 3' by its polymerase activity. Primer extension is performed at the temperature optimal for the particular polymerase that is used. Currently, the most popular enzyme for polymerase reaction PCR is Taq polymerase, the DNA polymerase from the thermophilic bacteria, Thermus aquaticus. This organism lives in a hot spring that can be near boiling and thus requires a thermostable polymerase. Okay, this is the process yang I kata tadi. So kadang-kadang kita akan buat 30 hingga 40 cycle. So you akan dapat banyak lah. 1 cycle you dah dapat 2, 2 cycle you dah dapat 4. Okay, for 3 cycles you dah dapat berapa banyak. So you can count lah. So yang ni dia denature, dia buka the bonding, dia anneal dengan primer je. Dia takkan lekat lah. Yang anneal tu primer yang boleh lekat je lah. Yang lain akan kat luar lah. So dia kena complementary. Lepas tu baru extension. Okay, dia akan extend one by one. Yang ni yang biru, hijau, merah, hitam ni adalah GACT lah. So yang akan tambah one by one. Okay, it's a nice picture. So this is how it happens lah. First cycle dia jadi 2 template. Then second cycle 2 jadi 4, 4 jadi 8, 8 jadi 16 until 35 cycle. So 2 to the power of 36. Okay, you will get 68 billion copies of the template. Okay. Okay tak so far? Faham tak? Hello? Hello?
Okay.
Okay, nanti pergi baca balik. So this part is important. Yang ni dah keluar jap. Saya tukar ni balik. Okay, we are back. Lagi dah 3-4 slide je, so we can finish. Okay, application of PCR. Amplifying DNA for cloning or analysis. So, you nak jadikan dia banyak untuk buat cloning in future. Such as in criminal investigation kan. You tengok kan macam forensic science and so on kan. Crime scene investigation tu semua kan. CSI semua tu. Criminal investigation and fossil analysis. Same compound could be found in the few herb species. Okay. Dalam few herbs, same compound you boleh jumpa. The similarity of gene sequence encoding the enzyme involved in this compound biosynthesis could be highly similar. So, dalam membuat compound dalam plant tu menggunakan DNA sequence juga. So, mungkin compound yang sama dalam plant yang lain, gene dia hampir sama. So, mungkin dalam satu plant you dah jumpa gene tu, another plant ada compound. So, gene dia akan hampir sama. Similar tau. Dia mungkin ada 1 or 2 nucleotide difference. Macam tu lah. Okay. The similarity of gene sequence encoding the enzyme involved in this compound biosynthesis could be highly similar. Thus, primers can be designed using the identified sequence in order to identify gene sequence of enzyme encoding similar compounds in other herb species. Okay. Testing for the presence of specific DNA sequence and comparing the DNA molecules. Okay. So, you nak test for ada tak certain DNA, then you compare. Okay. And then another one is quantifying DNA or RNA lah. So quantify means how much they are expressing. How much a specific nucleotide sequence is present in a mixed sample. You boleh buat, selain daripada buat qPCR, dia ada quantitative PCR. qPCR kita panggil dia. Dia tengok how much the gene ataupun DNA is expressed. Express kalau sometimes gene ada yang tak express. So how much this expression level of certain genes that synthesize express. Kalau dia punya function lah. Dia express kalau gene tu tak diam. Gene tu akan buat translation, transcription, dia akan buat mRNA, lepas tu dia akan buat protein. Ada gene yang dia senyap je. Mungkin kena dia tak trigger ke apa, dia tak ada function. Kita pun tak tahu apa function dia. Okay. Synthesize specific enzyme for specific compound production can be analyzed. Okay. So, chemical structure of DNA lah. Tadi dah belajar dah kan. Okay, done. Okay, yang tu je lah. Okay, ini apa? PCR in COVID-19 test. Tak ada apalah. PCR tests are used to directly screen the presence of virus, ada DNA, RNA kan. So kita hantar PCR pasal itulah. Kena kita nak check RNA tu betul ke tak. So mereka dah tahu RNA tu apa. So kita banyakkan dia. So kita boleh buat pembacaan. Kalau disikit, orang tak dapat kesan. So mereka ambil sampel kan. Mereka swab and take the sample. They send to the lab. The lab akan, dia kan buat PCR kan. Dia akan jadikan dia berbilion-bilion. So that when they check back, lepas tu kita akan run gel. Gel maksud dia akan tunjuklah band tu betul ke tak. Ataupun kita, so we will know betul ke tak. Which will be detectable in the body before antibodies from or symptoms of the disease are present. This means the test can tell whether or not someone has the virus very early on in their illness. Okay. All these things not very important. Tak keluar pun dalam exam view yang COVID-19 test. Okay, we are done. Okay ke?
Okay, Doktor.
Okay, go and read back. So kita ada one more class. We start at 3 o'clock lah. You'll finish by 4:30.